This is your typical diagram for explaining how DNA is packed within a chromosome. Of course, you've got your nucleus and cell up there at the top. Shows a chromosome with a, if you look down at that right arm, it's untangling, and when it gets done uncoiling, what you have is a single strand of DNA. What I'm telling you right here and right now is that that is completely and utterly wrong. And I'm going to show you in this movie why that is so. Before we get into this movie any further, I need to explain what you're looking at in this particular slide. Now this image, it's called a cell nucleus, highly diagrammatic, and it has these little compartments within it that look like everything's coiled up in there. And those things are called TADs. This is a mouthful, topologically associated domains. Okay, now what that means in plain English is that's a chunk of DNA all wrapped up, it shows there, but it has its own compartment within the nucleus. So there's some organization there that is recognized by the scientific community. Now, exactly how that is organized, they are yet to say, but they are saying that there is some level of compartmentalization within a nucleus, like our nuclei. Now, I'm calling it a compartmentalized degenerate endosymbiont, and I don't really want to get into what that means right now, but we'll talk about that a little bit more later. Now, that's just a diagram like the other diagram. I'm going to show you reality, something that is not a diagram, it was found under a light microscope. We'll talk more about these two images later in the movie. So if mainstream science now assumes that the nucleus is composed of these irregularly shaped compartments, each one has a different color so that you can distinguish them with all this DNA raveled up in there. If we are to assume that that's correct, then let's take that old traditional model and paste this diagram right on there and assume that that is indeed the way it is, and I think there would be a consensus about that as well. So let's go back to that diagram and remove all those squiggly lines and just look at the focus on the compartments themselves. If you'll notice, they're very irregularly shaped. That's very similar to what an amoeboid shape, a protozoan. I want you to think about each one of these things as their own cells within the nucleus. And if that were the case, these would be called endo, which means within, symbionts, which means very similar to something like a mitochondria or a chloroplast. They are doing something within that nucleus that is unique to their functions that helps the cell, our kind of cells, to develop and become all different kinds of cells. And each one of these TADs, or domains, will have some function in doing that. They are coordinated and working together for the greater good of the whole. So let's just paste this diagram on top of that traditional diagram in place of the other one. Now, let's check out a new diagram. This one shows all of these domains, TADs, I call them endosymbionts, separating from each other. Now, what would cause something like that? Well, the cell is beginning to break up under some kind of external stress or internal stress, and these things are beginning to dissociate from one another. I'm going to slap that on that old diagram that we were looking at, see what that looks like, and then we're going to compare that to an actual photomicrograph. This is a photomicrograph that you saw it's been cropped. You can see that these things are beginning to dissociate. They're beginning to separate from each other and spread out from the central core. And there's still a lot of organization here. But this is what's really going on. This is not some hypothesis. You're looking at it. It's actually happening. And so let's slap that on that diagram. Now we have another diagram of those amoeboid shapes, but they are surrounded by greenish looking circles of various sizes. Those circles are being ejected from those amoeboid shapes, which we will show with another photomicrograph coming up. So let's slap that onto that diagram and see where we go from there. Here's an actual photomicrograph of those circles escaping from that inner core. We're going to slap that on there also and see what that looks like. So here we're back with the original traditional diagram that we started out with, but we're saying at this point that all of this needs to go away, and that chromosome is not a good example for explaining what goes on inside that nucleus. So what we're going to do now is start from scratch, and we're going to just look at that chromosome all by itself. There it sits alone. And I'm going to compare that to some other diagrams that I came up with 30 years ago. But in order to do that, I'm going to have to invert that fella through CGI magic. So I've inverted him so I can use him to compare to the next illustration, which is this one. Now you're wondering, what in the world is all of that? That's very diagrammatic. That is my diagram from my early years. In order to really understand that, we're going to have to just bear with me and plot on through this. But you see those arrows? That second arrow, it's pointing to the centromere. So what you're seeing in the centromere is where all those chromosome arms connect. The big ones and the little arms are connected at the centromere. And my diagram is showing a lot of little black, looks like 4-leaf clovers or something, a big box full of them. And that would be where the centromere is, and that would be highly repetitive DNA. But we don't want to go into a lot of detail about that. So let's just move on to the next slide. And you see that I take a portion of that chromosome, that big black arrow, and it magnifies it, and it removes all the proteins that are causing those little black clovers and just showing the DNA itself. You see a lot of paddle fan shapes, and they have little circles attached to them, and they're attached to an even bigger, it's not a loop, it's a circle. These are all circles, but they're attached together. Now when we go to the next diagram, I'm going to circle one of those paddle fans that have the little, that are decorated in the middle. You can see it right there in the big red circle. When we go to the next diagram, that is pointing to all those circles, those different size circles. You got about 3 different sizes. You got a middle circle, you got 4 big circles surrounding it, and then you got all those 8 little circles, and they are all dissociated from that paddle fan that's encircled in red. So the question is, how do they do this dissociation? Well, that's a process that goes beyond the scope of this movie, and I would strongly recommend, if you are interested in learning more about this, that you head over to evolution4.wordpress.com and shop around and see what's there. And if you have any questions about anything, just send me an email, and I'll do my best to help you out with it. But this is a very important topic, because we need to understand how the DNA is put together if we're ever going to truly conquer genetic diseases like cancer. Thank you for watching this.